Welcome, Brookings Biology students. This is Mrs. Rydell, and today we're going to talk about some molecules that are important for living things and what those molecules do, where we get them, what do they look like, and what functions they have inside cells. We're going to start by making a list of 4 macromolecules. Remember, macro means big, and these are all 4 of these groups are molecules that are very important for living things. So we're going to make a list, and then we're going to talk about them and give you some examples. So number 1, carbohydrates. Number 2, protein. Number 3, lipids. Number 4, nucleic acids. If you look at that list, some of those words are familiar. We think of them when we talk about food groups and getting a balanced diet. Things like carbs and proteins in our food. We usually don't talk about lipids as a food group, but we do sometimes talk about those examples of lipids that we find in foods like fats and oils. And nucleic acids, we don't really think of that as a food group, and the reason is that nucleic acids include molecules like DNA. And remember, DNA is an important molecule, and it's one of the characteristics of living things is that all living things have a universal genetic code. They have DNA inside of them. And so if you're eating something that was once alive, it's going to have nucleic acids, DNA in its cells. And so we don't really have to worry about getting enough DNA because it's in all the foods that we eat.
We're going to start out talking about that first group of macromolecules on our list, which is carbohydrates. And carbohydrates are macromolecules. Remember, these are the 4 big molecules now found in living things. Carbohydrates contain carbon, hydrogen, and oxygen, those first 3 atoms that were on our list of important atoms for living things. And other kinds of molecules contain those 3 atoms as well. We see them in DNA, we see them in lipids, we see them in proteins. So what makes carbohydrates different from those other kinds of molecules is the ratio of carbons, hydrogens, and oxygens in our molecule. So if you look, carbohydrates always have a ratio of 1 carbon to 2 hydrogens to 1 oxygen. There's always twice as many hydrogens as carbons and oxygens in our molecule. And the simplest kind of carb is what we call a monosaccharide. Remember, mono means 1. We had that word before, monomers. Mono means 1, and the word saccharides comes from the Latin root word saccharose, which means sweet, one sugar molecule. These carbohydrates are kinds of sugar molecules. And so monosaccharides are molecules that have just 1 sugar. Example would be glucose. Glucose is a sugar. A lot of sugar names end in that ending, O-S-E. And if you look, the chemical formula for glucose now is C6H12O6. Remember, it's telling you what kind of atoms, carbons, hydrogens, and oxygen, and how many of each kind. Notice there's 2 times as many hydrogens as carbons and oxygens. There's 12 hydrogens, only 6 carbons, only 6 oxygens. If you look at a picture of glucose here, you'll see, remember, carbon can make those cool ring shapes. It can make chains. It can make all kinds of different shapes. But glucose is a ring-shaped molecule. And if you look in the ring, you see C for carbons. You have carbons here. Carbon, carbon, carbon. You have hydrogens, some hydrogens attached to those carbons. You have some O's. Here's an oxygen. Here's an oxygen. Here's an oxygen. If you count up the atoms that are in that little diagram, you'll see 6 carbons, 12 hydrogens, 6 oxygens. There's twice as many hydrogen atoms as carbon atoms or oxygen atoms. And that pattern that you see 1 carbon for 2 hydrogens to 1 oxygen. We see that in all molecules that are carbohydrates. Some important monosaccharides, 1 sugar, remember mono means 1 sugar, would be glucose, for example. And remember the chemical formula for glucose is C6H12O6. This second little picture is showing you another kind of sugar. Still has that 1 to 2 to 1 ratio. It's called ribose, and ribose is an important sugar. It's part of our nucleic acid group family of molecules, and we're going to talk about that in a little bit. But ribose is also a carb. It's a kind of sugar. And here's the chemical formula for ribose, C5H10O5. Notice it still has that ratio of twice as many hydrogens compared to the carbons or the oxygens. Sugars can have different numbers of carbons in them, but they always have that ratio. This is a really tiny little sugar. It's called glyceraldehyde, and the chemical formula for glyceraldehyde is C3H6O3. Still has that 1 carbon, 2 hydrogens, 1 oxygen ratio. All of these are simple sugar molecules, what we call monosaccharides. The cell now can go on, and remember, we can join smaller molecules together to make bigger molecules. I can take some of those real simple sugar molecules, monosaccharides, and join them together to make a bigger molecule. So if you look, here's my glucose molecule, and you can see the carbons and the oxygens and the hydrogens. Remember, we said sometimes we want to know what's in the molecule, and we make a little stick figure to show how the atoms are hooked on. Sometimes I just want to know the shape of the molecule. So what this little picture is showing you is that this glucose molecule, this hexagon here, is a glucose molecule. And in this case, we're just looking at the shape of the molecule. This has carbons here and hydrogens and oxygens attached to the ring, but we're not really looking at those individual atoms. We just want to see the overall shape. So if I take this glucose and I hook it together with another glucose, another glucose, another glucose, I can make a really big carbohydrate. And think about what we talked about, chemical reactions. How could I take individual little pieces, individual little sugar molecules, and hook them together, make a bond, hook them together using one of the reactions that we talk about? Remember, we talked about pulling out a water molecule and making a bond. I can take simple sugar molecules like glucose and hook them together using dehydration synthesis. Remember, that's our chemical reaction. That joint synthesis means to join something together. And dehydrate, in order for that bond to form, we're going to pull out a water molecule. And what it does is allow us to make more, we call them complex carbohydrates. Little bigger kind of carbohydrate molecules made by joining more than one sugar molecule together. So if I take monosaccharides, one sugars, and join them together, I can get a bigger carb. Some examples of sugars, carbohydrates, that are made by joining more than one sugar molecule. This next simplest size of molecule would be to take 2 sugar molecules and hook them together. And in science, the word for, or the prefix for 2 is di, D-I, and it means 2. Disaccharides. We had monosaccharides, 1 sugar. Disaccharides contain 2 sugar molecules. So this is a sugar molecule, and this is a sugar molecule, and they're joined together. They're attached by a little bond here. And joining, disaccharides are made by joining 2 sugar molecules together to make a bigger, a little bit more complicated carbohydrate. Example would be what we call table sugar. Table sugar is sucrose. Notice that OSE at the ending again. It's a clue that it's a sugar molecule. Another sugar that you may have heard of is the sugar that we find in milk. Milk sugar is, anybody know? Lactose. Lactose, notice the os at the end, it's a sugar molecule made by joining 2 monosaccharides together, and it's found in milk. Some people, maybe you know someone who's lactose intolerant. They don't have the ability to break down and digest that milk sugar, the lactose. We're going to talk more about that in our genetics chapter. Another kind of carbohydrate, more complicated, that we can make is, instead of joining 2 sugar molecules together, join a few sugar molecules together. The prefix oligo means few. And so oligosaccharides, ooh, that's a typo. Let's fix it. Oligosaccharides, D-E-S, not E-D-S. Oligosaccharides are made by joining a few sugar molecules together, like between 2 and 10. And oligosaccharides are important parts of molecules that are found in cell membranes. So if you look at these little kind of pinkish, these are cell membrane proteins, and attached on the surface are lots of those little hexagons. Each one of these is a little sugar molecule that's attached to the surface of the cell. And you can see them here. They're on the surface of all cells in your body. We're going to talk more about how they work in a minute when we talk about functions. But joining a few sugar molecules, maybe up to 10, make molecules called oligosaccharides, and oligosaccharides are found in cells as part of what we call a glycoprotein molecule. It's a combination, part sugar, that's the glyco part, and part protein. And we're going to talk about that in a second of what those do in cells, but they're important in cell recognition, helping the cell to recognize foreign invaders. I can take even more sugar molecules and hook them together. Remember when we talked about taking monomers and making polymers? This would be a good example of a polymer. Each of these little hexagons is a sugar molecule. And I can take those little monomer subunits and make a polymer, or in this case, since we're joining sugar molecules, I'm making what we call a polysaccharide. Poly means many. And polysaccharides are made by joining many sugar molecules together. We're talking hundreds to thousands of sugar molecules. These guys are pretty big molecules compared to just a simple glucose molecule. But each one of those little hexagons is a glucose. And so joining them together in different ways can make different kinds of polysaccharides. We're going to learn 3 important polysaccharides. And we're going to make a little list here. One of them is starch, found in plants. The other one is called cellulose, also found in plants. And the third one is important for animal cells, and it's called glycogen. And you store glycogen in your liver, in your muscle cells. It's a molecule for storing energy for later. If you look at these now, they're all made by joining many sugar molecules together in long chains. Remember we said carbons can make long chains, they can make rings. In this case, they're doing both. They have rings of carbons to make the glucose, but then the glucoses are all hooked together in a big long chain. So look at these 2 molecules here, because they're still sugars hooked together, but they're very different from each other. They look different, they act different in cells, they're different stuff. So what's the difference? Take a look at those pictures and see if you can see a difference between them. What's different about how they're put together? If you look, in this case, every other sugar molecule is kind of hooked in upside down and backwards. And so we're still joining sugar molecules, but we're joining them and putting them together in different ways to make these different substances. Look at glycogen. Glycogen is a very branchy polysaccharide. So we're still joining glucoses, but the way we join them together can make different kinds of polysaccharides.
So let's talk a little bit about carbs. And remember, carbs are coming from our food. Things like bread, cereal, rice, crackers, pasta have carbs in them. The very first, most important thing that carbs do for you is they supply energy. If you think about your body as a car, glucose is your gasoline that makes you go. Cells burn glucose for the energy that they need. Think of all the things you do that require energy. Running, moving your muscles, transport inside your body, cell division, repair, reproduction, growth, all of those things we talk about that living things do require energy, and it comes from burning glucose. Those polysaccharides now also have a function, and this one, glycogen, is the way that animal cells store their sugar for later. So when you eat a big meal, some of that sugar gets used right away, but usually you eat way more than you need at the time, and that sugar molecule, those extra sugar molecules, are stored in your body as glycogen. If you are an athlete, you know that one of the things coaches tell athletes to do before a big game is to carb load. What you're doing is taking in carbohydrates, complex carbs, we call them polysaccharides, like we eat pasta or pancakes or potatoes before a big game. And what you're doing is loading up your cells with stored sugar. Remember, glycogen is made out of glucose, 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 glucose. And so what you're doing is loading carbs. You're storing up sugar in your sugar bank as glycogen. We store it in our muscles, we store it in our liver, and what you're doing is saving some of those glucose molecules for when you're going to be running down the soccer field for 45 minutes or charging up and down the basketball court or running track or whatever you need energy for the next day. It's stored as glycogen in your cells. Plant cells store their sugar a little bit differently. If I'm a plant cell, plants store their glucose as starch. You know, think about all the foods that you eat that have starch in them. Think potatoes, pasta comes from wheat. All of those foods that we eat with starch in it come from plants, and that's because that's how plants store their sugar. When they do photosynthesis and they're making glucose, right, they're making sugar for themselves, some of that sugar gets stored for later. Glucose, 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 we hook it together and we make starch. The plant saves that for later, like putting it in a tool of potato. And that way the plant has extra energy for later on when it's wanting to do plant things like grow and reproduce and transport and all the things that plants do. The third molecule that we talked about is this molecule. Plants also make something called cellulose. Remember that every other one is upside down and backwards. Okay? Cellulose is a molecule that makes plants sturdy. Makes plants sturdy. If you remember watching a building go up before, one of the first things they put down before they add the bricks and the walls, they put up those big metal I-beams. They're support beams. Right? They anchor the building, they give it support, and that's what cellulose does for a plant. Think celery. If you've ever eaten celery and you break the stalk and those little strings that run down the celery, that's cellulose. Comes from the same thing, cellulose, celery. They're building blocks. They're making it so the celery stalks will stand up straight and give them strength. It makes them sturdy. So cellulose isn't a way to store sugar for later. It's a building block for making plant stems and be able to stand up and be sturdy. Okay, other things that we use carbs for. One of the things we build with carbohydrates is this molecule. It's called a glycoprotein. Notice it's a combination molecule. Part glucose, glyco, part sugar, and part protein molecule. And it sits in cell membranes. One of its jobs is to sit in cell membranes, and it's on the surface of every cell that's in your body. If you look, the protein part anchors in the cell membrane here, and sitting on the surface, look at those little hexagons again. They're sugar molecules. Sugar, sugar, sugar. They make a pattern, and everybody's glycoproteins are different. So their job is to sit and stick on the outside of cells, and they're on all the cells in your body. They're on blood cells, they're on skin cells, they're on any kind of a cell that's in your body has glycoproteins. So they're proteins that have sugar molecules attached to them. They're sugar molecules attached to them, and they sit on the surface of cells, and their job is to recognize me, self. Are you like me or are you different from me? Why is that important? Because if something gets into your body that's not you, it's probably something that shouldn't be there, like a germ or a parasite of some kind. What those glycoproteins do is they help you to recognize something that's different from you. Why is that important? It's important in blood typing. Some of you may know what your blood type is, like I'm O positive, and someone else is B negative. You may have heard someone talk about blood types. When you say someone is A or B, positive or negative, what you're saying is what kind of glycoproteins they have. So someone who is A-type blood has different glycoproteins on their cells than someone with B-type blood. It's important for recognizing you're like me. It's important in organ transplants. If you think about a transplant, if you're going to get a new liver or a heart transplant, the first thing they do before they do the transplant is they got to find you a donor match. And what they're matching, what they're looking for a donor that has glycoproteins that are similar to you. Because if you give someone the wrong type blood, or you give someone an organ that's not a donor match, your body will see that as that doesn't belong, just like it would recognize a germ that doesn't belong in your body. And it can lead to your body rejecting and attacking that organ if it's the wrong match. And that has to do with what kind of glycoproteins. We're matching glycoproteins to find you a donor match. It's important in recognizing germs. Germs have different glycoproteins on their surface than we do. So those glycoproteins on your cells are looking for a match. Are you like me or are you different? And if you're different, it's probably because you don't belong. We're going to stop right there. Welcome, Brookings Biology students. This is Mrs. Wright.